So, good afternoon, guys. I hope my slides are visible to all. And today we'll be completing the last lecture of the course Cell Culture Technologies, and we'll be discussing the concepts that we have learned in this week. Okay, so we'll start. So the first question is Stoney's equation is used to quantify the dash generated in molecular thin films from different molecular recognition events. So the options are energy, bulk stress, surface stresses, or density. Can anybody guess?
Surface stresses, option C.
Okay. Any more answers? So yes, the correct answer is surface stresses. So what happens, the basic principle of Stoney's equation is, so it applies to thin film materials. So for example, this is the thin film material, and whenever this film is bent or forces being put on this particular film, it will bend. Okay, it will have this kind of conformation. So the radius that with which it will bend, this particular force or this particular stress will be identified using this Stoney's equation. Okay, and this has been applied in various biological aspects also, especially studying the muscle stress or the muscle force generated. Okay, so this is the basic equation that we use. So if there is any doubt, you can ask. So let's move on to the next question. So next question is on a cantilever, which of these cells will generate a higher force? So the options are cardiac cells, brain cells, skeletal cells, or liver cells. What can be the answer? So in our body, when we consider our human body, so there are only one force is like there is only one tissue that generates the force, that is the muscle. Okay, and muscle are of three types mainly. One is smooth muscle, one is cardiac muscle, and the last one is skeletal muscle. So smooth muscle is basically present in the inner lining only. Cardiac muscle present only in the heart, and skeletal muscle present on the bone and all those stuff where the actual force is needed. So if we consider the force of these three muscles, the force generated by the skeletal muscle will be the highest, then the cardiac muscle, and then the smooth muscle. Okay. So smooth muscle cells are not in the option. So the this is like pointed out. And considering the brain cells and liver cells, they are like the fourth generation is minimum in these cases. So the correct answer will be skeletal muscle cells rather. Okay, so the option is C, skeletal muscle cells. Okay, so let's move on to the next question. So which of the following parameters from the cantilever detection system is the most important to determine the force or stress on the myotube? So the options are modulus of elasticity of the material from which the cantilever is being made, the angle of the beam deflection, position of the myotube, or intensity of the beam. Can anybody guess? So here the answer is angle of beam deflection. Okay. And this is the process that I was talking about. So what happens is here, as you can see, this is the stage where the muscle is kept or the myotube that we are talking about. So here it is kept and here it will be stimulated using any of the means, using any electrical means or mechanical means. Okay. Now, this is the cantilever uh, on which the myotube has been placed. Now, this is the microscope objective which you can see and place the cantilever. Okay, because this will be a small cell, so we require some like visual machinery also to place the sample very carefully. Now, what will happen? This is the electrical stimulation system. Okay, because the electrodes are being placed here. Now, we put a laser beam on this, and then this beam will be deflected from this side. Okay. Now, the angle of deflection, which will measure the force that is being generated on the myotube or the stress that is being generated on the myotube. Okay. Using these particular equations that we see, these particular equations belong to the Stoney's equation. Okay, so using these particular equations, we generally calculate the force or the stress on the particular muscle that we see. And there are also some properties like the Young's modulus or the Poisson's ratio of the cantilever, that the material that is being used to make the cantilever, and some of the elasticity properties of the myotubes also. So these are the constants that we keep in these equations and we calculate the value of the force or the density that we or the stress that we want to ask or the stress that we have to calculate of this particular muscle cell. Okay, so this is how the system works. This is the cantilever-based system for detection of force on a particular myotube. And these are the particular equations which helps in calculation of that particular force using the angle of the beam deflection. Okay. And how what happens, for example, like we discussed in the first question also. So suppose this is the muscle cell. Okay. And here we are considering as a thin film. Hmm. Now we are putting the beam here and this is the theta by which the beam has been identified. Okay, now we put the muscle into some stress, so it will be bent. Okay, now the muscle is being bent. So now when we will put the beam, the angle of deflection will be, suppose it is theta prime, so it will be different. It can be more, theta prime can be more than theta, or theta prime can be less than theta. Okay, depending upon whether it is relaxing or contracting. So now these theta values that we get from this particular system, we just put in these values and then we can calculate the force from these particular equations. This is how we calculate the force or the excitability of the particular muscle cell. And if you guys are interested, you can go to these particular references and you can read more about these equations and how this particular instrument functions in detail. Okay. So if you have any questions, you can ask. So if not, then we'll move on to the next question. So DASH is a major neurotransmitter present in the neuromuscular junction. So options are serotonin, glutamine, acetylcholine, glutamate. Can anybody guess? So here the correct answer is acetylcholine because this neurotransmitter is majorly found in the neuromuscular junction. And this is the particular diagram of the neuromuscular junction. So as you can see, this yellow thing is a motor neuron. Okay, all this is the perfect diagram of a motor neuron, and this is the muscle cell. Now, like for example, the skeletal muscle cell, when we have to move a muscle or we have to move a hand, so our brain will send the signal to the muscle. Okay, so sorry. So the brain will send the signal to the muscle, and what will happen? This muscle will have a connections with the neuron. Okay, so this neuron will send a signal to the muscle in the form of passing through the neuromuscular junction. So what the neuromuscular junction does? So it transfers the signal from the neuron to the muscle in the form of chemicals. Okay, that we call neurotransmitters. So these are the chemicals that this particular motor neuron will release, and majorly these chemicals will are acetylcholine. Okay, so the muscle has different acetylcholine receptors. So whenever this particular signal comes, so acetylcholine attaches to the receptor and it further increases the downstream signal. Okay, so this is the basic mechanism how a neuromuscular junction works. And there are various diseases associated with it. So whenever this particular signaling or any of the receptors are hampered, then the process of transmission also gets hampered. So that is the cause of various diseases. So this is the thing with the neuromuscular junction. So let's move on to the next question. So the acetylcholine receptors are divided into two types based on the type of ligand it is activated with, type of toxin it is inhibited with, the function it performs, or none of the above options are correct. So can anybody guess?
Answer A, the type of ligand it is activated by.
Okay. Any more options? So here what happens is like when we are talking about acetylcholine receptors, so they are categorized into two classes. Okay. One is metabotropic and the other one is ionotropic. And the second name for this is actually the complete name will be metabotropic muscarinic acetylcholine receptors. And for this one, it's inotropic nicotinic acetylcholine receptors. So these particular receptors are named based on their function as well as on the ligand it is bound. Okay, so this is the function that they are performing, and this is the name of the ligand that they are being bound to. So option A and C most both can be correct in this case. And what happens in case of metabotropic receptors, the downstream signaling is the CAMP or CGMP pathway. Okay, this comes in the metabolic profile of the body. And in these are the ion channels that basically excites or excites the cell and transfers the action potential to the muscle. Okay, so this is basically the thing that want to discuss with you. So the option can be both A and C. Okay, so does anybody has any questions in this case? So if not, then we'll move on to the next question. So you are generating a neuromuscular junction in vitro, and what will be the sequence of adding the cells in the culture? So the options are first you'll add skeletal muscles, then fibroblast. First you'll add motor neurons, then skeletal muscles, or first you'll add skeletal muscle, then motor neuron, or you will add both at the same time. So can anybody guess?
Section Answer B, motor neuron, skeletal muscle.
Okay. Any more answers? So what happens when we have to create a neuromuscular junction in vitro? So what we'll do, we first isolate the skeletal muscles. Okay. And at what time we isolate the skeletal muscles? Can anybody answer? We have discussed in the previous lectures.
Isolate the satellite cells.
I'm asking about the timeline. When we isolate the skeletal muscles or the satellite cells from the embryonic rat.
Embryonic 7 or 8.
Sorry, can you repeat?
Embryonic 7.
No. We isolate the muscle cells at embryonic 18. Okay. And what we do is we isolate the satellite cells, and along with that we have the fibroblasts also. So now we have to grow them. So first we add the skeletal muscles with the fibroblasts, and then we add the basic FGF. Okay, this we have to do in very little quantity so that the skeletal muscles can proliferate. Now what we do after this whole thing is done, we just take out half of the media. Then we add the motor neuron. Okay, so what is the age that we isolate motor neurons is? It's embryonic 14. Okay, from the ventral horn of the spinal cord. So, and now what we do after few days, we'll remove the full media and that is there, and we add the new media. And further, after few days, we can check the with various functional tests that the particular neuromuscular junction has been formed or not, and it's functioning or not. Okay. So with this particular flow chart, we generally develop the neuromuscular junction. Okay. So the correct answer is option C. So first we have the satellite cells, then we add the motor neurons. Okay. So, can anybody think about why is this particular uh, format of adding cells in a culture is? Just think about it and we can discuss now. So actually, these what we see is the development pattern of the basic human body. Okay. So as we can see from this image also, I'll remove this. So as you can see here, these are the time points. Okay, sorry. So these are the time points at which different scenario that takes place in the development of the neuromuscular junction. So first, what happens? There is development of the myofibril or the myotubes, and there is generation of the acetylcholine receptors on the particular myotubes. So this is the first step in the development. Then the second step is there is the development of the motor neurons. So the axons of the motor neurons, they reach to the muscles so that they can link with the different acetylcholine receptors. Okay. Now, what happened? The in this point when there is parturition or the birth of the baby, so at that particular time there is some muscular junctions has been formed depending upon the synapses that we see, and the particular acetylcholine receptor is being linked to the axon or the axon of the motor neuron. Now, after birth, in the adult motor neuron unit, we can see different motor neurons that is connected with the acetylcholine receptor in the muscle. So at after P14, after birth at P14, we just see the distinct neuromuscular junctions, but in this case, PD multiple axons like connected to the muscles at the same point of time. Okay, so here we see distinct uh, neuromuscular junctions. So this is the pathway that is being followed by the in the development pattern of neuromuscular junctions. That is why when we add in vitro, so first we add the skeletal muscles. So we allow them to develop. Okay, providing different growth factors as well as we have some amount of fibroblast also there. Then after that, when the particular growth is there, so that we can check with the help of different antibodies or different functional assays, and then we add the motor neuron. Okay, so in the same case, we'll apply for the motor neuron also. So when we add the motor neuron in the culture, they will also be at in the bilkul, primitive state. So then we allow them to grow. And then after that, when we also add some of the growth factors in this that do not hamper the muscle cell. Now, when we have this motor neuron. So after that, we allow both of them to grow together, and then it will form a neuromuscular junction. Okay. So anybody has any doubt in this question, can ask. So if not, then we'll move on to the next question. So curate is an example of a dash muscle relaxant that blocks the nicotinic acetylcholine receptors, one of the two types of acetylcholine receptors at the neuromuscular junction. So can anybody guess?
Answer 2, deep rising.
Okay. Any more answers? So the correct answer is option A. It's non-depolarizing. So what happens is, as you can see in this particular diagram, This is the particular diagram depicting the neuromuscular junction. I have already shown you this, but what happens in this case when we have a any agent that blocks the uh, transmission of signal from the neuron to the muscle cell. So this is particularly muscle cells, and these are the synaptic presynaptic neuron. So what happens here? The acetylcholine that is shown in yellow will bind to the acetylcholine receptor. So this is the nicotinic acetylcholine receptor. So now, what will happen if we have this agent? Suppose, for example, Cure. So here it will bind. It is the competitive inhibitor of acetylcholine. Okay. So now it will bind and it will not allow acetylcholine to bind, and it will stop its function. Okay, so it will depolarize, sorry, it will stop the depolarization of the membrane, which acetylcholine does. So here it is non-depolarizing in nature. That's why. Okay, so this is the basic mode of action of curate that we talk about, and it is being used in various functions. For example, when we are using it is used in surgeries, basically as a muscle relaxant. Okay, so which do not allow any signal to pass through from the neurons to the muscles. Okay, so this is the basic function of curate that we talk about. Okay. So let's move on to the next question. So what is the chemical nature of curare? So is it a neurotransmitter, alkaloid, stimulant, or anti-anesthetic? So can anybody answer?
Option C, sir.
Okay. Any more answers? So curare is basically an alkaloid. Okay. So alkaloid are those compounds that have isoprene-like units. Okay. So this is the example of an alkaloid. Curare is an example of alkaloid. Just like morphine or cocaine that we see, it's also an alkaloid. Okay. It's not a neurotransmitter. It is not a stimulant. It is basically a relaxant. So it cannot stimulate anything, and it is also not an anti-anesthetic. Okay, so let's move on to the next question. So bungarotoxins are a group of closely related neurotoxic. Uh, like, what's the chemical nature of bungarotoxin? Is it a stimulant, lipid, protein, or neurotransmitters? Can anybody answer?
Option B, sir.
Sorry, option
B.
B. Okay, any more
Lipids.
answers?
Proteins.
Okay. So yes, it belongs to the class of proteins, and like it is basically a neurotoxin that is being isolated from different poisonous snakes and great. Okay, so the name is also on the scientific name of a particular snake only, and these are the proteins. that are nearly 73 amino acid long, and that are, the different regions are linked with the disulfide bond. So this is the basic structure of the bungarotoxins that we study, and they affect the neuromuscular junction only. So whenever a snake bite happens to a human, so in that case, the human gets instantly paralyzed, and it will not be able to move it himself or herself, because the junctions that the signal passes from which the signal passes through the axon to the muscle, that is stopped. So that's why the person is paralyzed in that case. Okay, so this is the story of Bangara toxin, and let's move on to the next question. So, alpha Bungaro toxin. So there are different kinds of Bungaro toxins that bind to, uh, like, what's the functionality of that particular toxin that is binding to the nicotinic acetylcholine receptors of skeletal muscle. So can anybody guess?
Irreversibly.
Irreversibly. Okay, any more answers? So the correct answer is yes, irreversibly. It binds irreversibly to the nicotinic acetylcholine receptor. So as we talked about, there is muscarinic and nicotinic receptors. So when we label any acetylcholine receptors, so for muscarinic we write like this, and for nicotinic we write like this. Okay, so this is the case, and if we talk about the different types of bungarotoxin, so this is alpha, beta, gamma, and kappa. Okay, so alpha basically binds to the neuroacetylcholine receptors of neurons as well as muscles. Okay, the main target is muscle here. Fine. Now, when we talk about the beta and gamma, so they basically have the tendency to increase the acetylcholine receipt. That means more excitability. Okay. And for the kappa one, it basically binds to the neuronal nicotinic acetylcholine receptor. Okay, so this is the basic difference between these four kinds of bungarotoxin, and they are released from different kinds of snakes also. Okay, so if anybody has any doubt, you can ask. If not, then we'll move on to the next question. So yes, this is the major function of the bungarotoxin. So it basically acts on the muscle and deactivates the acetylcholine receptors. Okay, so there is one more neurotransmitter, glutamate. So what is the nature of the glutamate? Is it inhibitory, excitatory, a lipid-based, or a protein? So can anybody guess the correct option? Hmm. Sorry, can you repeat?
A.
Okay. Any more answers?
A. Inhibitory.
Okay. So the correct answer is option B. It's an excitatory neurotransmitter, and this is the basic uh, function that we talk about. So when we talk about glutamate, so it basically excites the postsynaptic neuron and increases the action potential or the opening of channels in that case. So it excites very much the postsynaptic neuron. Okay, so this is the case of the glutamate neurotransmitter. And it is not a lipid or protein based, it is an amino acid. Okay, so that will not go into these two categories. So that's why this is the case. Okay. So the next question is gap junctions are common feature of which kind of cells? Is it the cardiac cells, brain cells, skeletal muscle cells, or liver cells? Can anybody guess?
Cardiac cells.
Option A.
Okay. Option A. Yes, it's the cardiac cells. This we have discussed in the last lecture also. So when the cardiac muscles are like we talk about the cardiac muscle cells. So if this is the cardiac muscle cell, so these are very much linked to each other by the gap junctions very tightly. Okay, so that they cannot move with there is no flow of the cell, the flow of blood through the cells. Okay, so next question is dash is the process of patterning a substrate. It is wet etching, photolithography, dry etching, or plasma. Can anybody guess? Okay, any more answers? So what we do is like in these cases, uh, the correct answer is photolithography. So what happens is suppose there is a substrate on which a particular pattern has to be formed. So what we do is first we put that particular pattern on the substrate and then we use either UV rays or any other kind of light, like infrared rays for putting this particular pattern on this particular substrate. Okay, we put that for a very long time and then we keep it for development. Okay, now when we kept it for development, so now this substrate will have a additional pattern of this particular uh, thing that we wanted to develop on the substrate. Okay, and this is the major like tool that we use for use on for generation of various nanoparticles on a particular substrate. Okay, or any other nanoparticle layer for to put on a particular substrate. For example, in COVID-19, there were some masks with the coating of antibacterial or antiviral coating was produced. So these were produced by the process of photolithography only. Okay, so this is the case. If anybody has any doubt, can ask.
So can you repeat explanation?
Okay, so what happens is, suppose there is a substrate, okay, on which we have to make a particular pattern of a particular thing. For example, nanoparticles only. Okay, so what do? We keep the substrate and then we keep the layer of the nanoparticle above it because we have to develop that particular layer on the substrate. Now, from the above, we put a particular wavelength of light, that is, generally we use UV light for that, to transfer this particular pattern on the substrate. Okay. So after the develop, because that's why it is called photolithography. Okay, because we are using light. So now what we do, we keep it for some time for development, and we see the particular substrate had this particular pattern of the nanoparticles. Okay, this is the basic procedure of photolithography that we use. And this is generally used in case of our biological terms. It is being used in developing various nanoparticles based things or various nanoparticles based coatings that we use. Okay, is it fine now?
Okay. Yes,
Hmm.
ma'am. Yes.
Okay, so let's move on to the next question. So you have done a primary culture of hippocampal neurons. You added glutamate for their development, and you found that after doing electrical excitability experiment that the neurons are not electrically active. What can be the most probable reason for the above stated problem? So the options are: the glial cells are outnumbering neurons, there is no serotonin in the culture, there is no skeletal muscle cells added in the culture, or there is no glial cells present in the culture. So can anybody think and tell the answer?
Option A.
Okay. Any more answers?
Option yes. Yes.
Okay. So what happens is, generally in this case, when we grow particular neurons, especially the hippocampal neurons, so when we are growing these particular type of neurons, we add some amount of glutamate in the medium. So what does glutamate do? They basically helps in the development of neurons. Okay, so that's why we add certain amount of glutamate in the particular neuronal culture. Now, as we know, that when we isolate hippocampus, so can anybody tell when can we isolate hippocampus from the rat? At which embryonic stage we isolate hippocampus?
E14.
Yes, we isolate hippocampus at E14. So in hippocampus, we will have certain kinds of cells. Okay, there will be neurons, interneurons. Yes. So interneurons or the oligodendrocytes, astrocytes, and various other glial cells. So oligodendrocytes and astrocytes are also type of glial cells only. So if we have to like culture only these particular neurons, so along with that, the glial cells will also be present in the culture. And why do we need glial cells also in the culture? Because they secrete certain factors that helps in the development of neuron. Okay. But the major problem arises when there are more number of glial cells in the neuronal culture. So if there are more number of glial cells in the neuronal culture, what will happen? They will basically absorb the glutamate. Okay, so they act as a sponge for the glutamate. So they absorb all of the glutamate from the culture that will not be able to, so the neurons will not be able to grow in that particular or develop in that particular environment. So if we do a electrical excitability test after some time, so what will happen as these have not grown properly, they will not be electrically active. Okay, so the reason will be because there are more number of glial cells in the neuronal culture. That's why they are not able to grow and that's why they are not electrically active. Okay, so anybody has any doubt in this question? So if not, then we'll move on to the next question. So this is a diagram of the growth curve of particular animal cells. Okay, so beyond what cell concentration the saturation cell density is achieved. So the options are 10 to the power 4 cells per ml or between 10 to the power 4 to 10 to the power 5 cells per ml, 10 to the power 5 cells per ml or 10 to the power 6 cells per ml. So just study this diagram and then answer.
Answer D, 10 raised to 6.
Okay, any more answers? So when we study this particular type of growth curve, so this is a typical growth curve of an animal cell in culture. Okay, so like every other bacterial cell we study, there is a lag phase, then there is a log phase. Okay, and this is the saturation phase. And after some time, it will be the death phase. Okay, so in the lag phase, there is very minimal amount of cells, as you can see in the image also. So what is the lag phase means here? When we passage cells or we add, for example, this is the culture plate, we add the cells here. We allow them to stick to the plate and like adapt to the conditions that they are in particular culture conditions. Okay. Now, when they are adapted, they enter into the doubling phase or the log phase. So here they'll double their number and increase in the number. Okay. So here what we'll do, the growth rate of the cells will be calculated in this particular phase, that is the log phase. And then there will be a particular time when these cells will be in a saturation. So what is the saturation in this case? So as we know that for considering this as the culture plate that we are studying in, so when we animal cells uh, are fully confluent, okay, so that means they have covered the full surface area of the plate. So now what will happen? Their dividing capacity will also be reduced. So in that case, they have been reached a saturation phase. Okay. So from this time, they'll like dividing will be decreased. And the plate that we are talking about, they will cover the whole surface area of the plate. Okay. Now, what will happen if we do not passage cells after this time? They will enter into the death phase. So they'll now start scraping on its own from the culture plate, and the dead cells will be start floating in the medium. Okay. So this happens in normal scenario, and this is the normal growth curve of this particular animal cell. So according to the diagram, as we see, if we have a saturation density of greater than 10 to the power 6 cells per ml, the saturation density is achieved. Okay, so the correct answer is option D. Fine. So if anybody has any doubt in this question, can ask.
So the confluency has reached 10 to the power of 5 Celsius.
You are talking about the confluency, right?
Yeah, yeah.
Yeah. So yeah, it can be reached at that particular value.
So how the answer will be 10 to the power of 6?
Like here, as you can see in this particular graph, this is the cells per ml and this is the cells per centimeter square. So I have asked in the values of cells per ml, okay, not in the area context. So when we talk about the confluency terms, we'll talk about in the centimeter square. But if we have to answer in case of ml terms, okay, for per media or per ml of the media, how many cells have been reached? So in that case, we'll use this much. So because we are talking about the density here. Okay, so that's why we have answered in
Oh,
this case.
okay.
Any more doubts?
No, sir.
Okay, so let's move on to the next question. So the commonly used media for actively dividing cells is Delbeco's modified Eagle media, ISCOV modified Delbeco's medium, or Roswell Park Memorial Institute 1640 medium, or all of them. Can anybody answer?
Commonly used is answer A.
Okay. Any more
Option D.
answers? And D. Okay. Any more answers? So the correct answer would be option D. Okay, because like when we talk about the DMEM or IMDM media, these have the constituents similar in nature. Only one or two are like different in this case. So these media we use for adherent cell culture. And these particular RPMI media we use for suspension cell culture. Okay, so there can be more differences in terms of the constituents of the media, but the basic constituents that we use in all of these mediums is there is but all the type of amino acids we have. Okay, now and second we have vitamins, salts, salts in case of or we need calcium salts, magnesium salts, sodium and potassium. Okay, these are the major ions that we need in our particular system. And then in the last, we have certain buffering agents. For example, we have sodium pyruvate or bicarbonate. And we have certain pH indicators also, for example, phenol red. Okay, this is the basic constituents that we have in these particular medias. And the difference can be like one or two constituents in different concentrations or in different or we have additional one or two constituents. But these are the basic media that we use in our day-to-day animal cell culture. Okay, and these are and certain antibiotics also we add in this while culturing. Now, one component is missing in this particular media components. Can anybody tell what can be that? I have not mentioned it yet.
Anti-anti, so
Hmm. Sorry, can you repeat?
anti-anti.
Hmm. I didn't understand. Sorry.
Antibiotic and antimicrobial.
Okay. No. Any more guesses? So for the development of animal cells, we add one more component that is serum. Okay, this is not added in the particular medias when as a constituent because of two things. When we have certain kind of experiments, we'll add it additionally. Okay, and these particular medias without serum are called incomplete media. And when we add serum, so like in our cell culture, we use FBS, so fetal bovine serum. And then after adding this particular serum to the media, we call it as complete IMDM or complete DMEM media. Okay, so this serum is particularly a source of growth factors and different hormones. Okay, that provide the nutrients to the growing cells. Now, there is the difference in concentration of this particular serum. Okay, so when we have we revive cells from liquid nitrogen, so in that case we add high amount of serum, basically 20%. And when we use this in normal growth conditions, then we add 10% of serum. Okay, this is the basic scenario that I'm telling about that we use in our day-to-day culture conditions. Okay, so anybody has any questions in this case? So if not, then we'll move on to the next question. So it's a calculation based question that we particularly use in our cell culture facility in like every day. So uh, we need to seed 30,000 cells in 24 well plate. Okay. So you chose 80% confluent HeLa cell line. So you have to seed HeLa cells in that particular 24 well plate. That is 80% confluent. So now you counted the cells using a hemocytometer and found that like using a hemocytometer, we'll count in 4 quadrants. So in each quadrant, there is 98, 96, 83, and 79 cells in the 4 quadrants respectively. So now we have to add the media in that particular cell suspension to complete the volume, and then we add in different wells. So we have to calculate how much of serum free media we need to add to complete the volume in each well. So please calculate this and we'll discuss. If you need any more information, then let me know. So one more information, like in a 24 well plate, when we add media plus cell suspension, so in each well, media plus cell will be equal to 500 microliter. Okay, so you have to calculate based on this volume. So did you guys come to any answer? Or shall we discuss? Okay, so let's discuss. So what we have is we have a 80% confluent HeLa cell culture. Okay, we'll not use any 80% in this calculation because this is only for our knowledge whether we have to seed the cells or not. Okay, so now what we'll do, first we remove the media. Then we add trypsin so that the cells will be detached from the surface. Okay, now we add certain amount of serum with the IMDM. Okay, and then we collect the cells in a falcon and spin it so that we'll get a pellet of the cells. Okay, now what we do, we dissolve it, this particular pellet in PBS, 1x PBS. So now we can make dilutions or we cannot, we do not make the dilutions. So generally, like in as a person, I do not make any dilutions. I like dissolve this particular cells in PBS, and then from that particular volume, that suppose that is 10 ml, I take 10 microliter and then I measure the number of cells in the hemocytometer. Okay. So from the hemocytometer, as you guys would remember from the previous lectures, in the hemocytometer, this is the big square that we have, and this is divided into four quadrants, which have lines like this. So this is the first, second, third, and fourth quadrant. So we count the number of cells in this particular quadrant, and then we calculate the total number of cells per microliter that is present in this particular 10 ml of the PBS. So how do we calculate? In this case, we have 98, 96, 83, and 79 cells. Okay. So the formula is 96 plus 98 plus 83 plus 79. Okay. We have calculated from four quadrants into the multiplication factor. So this will give us the number of cells per microliter. Okay, that we have in this 10 ml culture of 10 ml solution of cells plus PBS. Okay, now from this particular suspension, what we'll do, we'll again spin this particular solution. The cells will be pellet again, and then it will be dissolved in the same amount of PBS that we added in it. So we'll make up the volume till 10 ml, dissolve this particular cells in the media, and this will be the solution, that is media plus cells. Okay, now from this, what we'll do, this is a cell suspension that we are talking about. Now we have to add 30,000 cells per well into 24 wells. So that will be equal to 7,20,000. Okay, 7,20,000 cells in whole plate that we have to seed. Now, what we'll do, we have to have this much cells. So if we divide by this many cells that we got from this particular scenario, so for example, we got M cells here. So we'll divide this by m. From this, we'll get the volume V microliter that we have to add in the cell suspension. Okay, so this V microliter from this particular 10 ml culture, we'll add in this, and then we have to add IMDM. Okay, so now how to calculate this volume of CIMDM that we are using it. So we have to calculate this particular volume of CMDM that we have to add in the final suspension. So I told you that each well will have 500 microliter of the culture like media plus cell suspension. So the total volume that we add in 24 wells will be 24 into 500. That will be nearly 12,000 microliters. Okay, so now for calculating the volume of media, we'll subtract this V from this. This will give us the volume of the IMDM or the media that will be added in the final suspension. And from that final suspension, we'll add 500 microliter in each of the wells. Okay, so now can you guys calculate, or do you have any questions in this particular question? Please ask if I need to explain it again.
So once it is checked with hemocytometer, again why to add that CIMDM media? So
Sorry, can you please
again,
repeat?
I will need to explain it.
I didn't understand it. Sorry.
When we already counted in hemocytometer, again why to add that CIMDM media again to skin?
Okay. So, like, uh, while we cannot add in the hemocytometer with the media. Okay. So here the cell suspension is in TDS. Now, when this we count these cells, we spin these cells and we add media. Okay, we cannot count the cell suspension with the media under the microscope. We'll not be able to get the clarity on that. Did I answer your question?
Sorry, sir, I'm still not clear. Can you repeat once?
Okay. So you are asking about this particular thing now, why we add 10 ml of CMDM after counting the cells, right? If I'm understanding the question correctly.
Yes, sir.
Okay. So what we do is first, uh, when we detach the cells from the plate, okay, so we add like PBS in that particular cell suspension to dissolve that particular suspension, okay, to make a homogeneous solution. Now, in order to like that serves two basic things because we have added trypsin we have old media in that, and we have to count the cells. Okay, so now we give a PBS wash. Okay, so we dissolve those particular cell suspension in PBS, and from that particular cell suspension, we actually make dilutions. Okay, we cannot make dilutions in media. So this particular step is for dilutions. Here I have not shown the making of dilutions in this case. Okay, so we make dilutions, then we add in the hemocytometer. So now what we do, because we have to plate them back, the cells in the culture plate. So we cannot add directly from the PBS to the media. So now we spin it down and we add complete IMDM medium so that the cells are in a suspension with the nutrients. So now what we do, we make different like if we have to add different plates, then we add directly from this, and you take this directly 1 ml from this and add in different culture plates.
Okay.
Hmm. So everybody understood the calculations, or shall I repeat once more? So after calculating this particular like question, we'll get the media in nearly 11.191 ml. Okay. So this particular media we have to add in the final cell suspension. So these calculations are used in day-to-day animal cell culture experiments. So when we have to add certain number of cells in a particular well plate, or we have to seed some certain number of cells, then we use these particular calculations. Okay. So you have to be very much thorough with all these calculations because these will be your day-to-day experiments. So you guys can like go and then practice it. And if you have any doubt, you can comment on the videos also. I'll be free to answer them. Okay. So let's move on to the next question. So commonly used media for actively dividing cells is, the options are MTTSA, annexin 5 staining, trypan blue staining, or all of the above. Can anybody guess? So here the option is all of the above. So all these tests, and there are many more tests that basically helps in. Sorry, the question is wrong. Uh, Here we are using for cell viability testing. Okay, So these particular tests are used for cell viability tests, and there are many more tests that are used for viability testing of cells. So MTTSA is the formazan assay that we use, and tetrazolium dye we use for that. So what happens? This is the basic principle of tetrazolium, and when the cells are alive, so what happens? They'll take up the tetrazolium and they'll reduce in and form the formazan, that is a purple colored compound. So this is the basic mechanism. It will use mitochondrial enzymes, for example, NADPH, and the reductase will, if the cells will be arrived, there will be reductase present. So the reductase will, what will do? They will transfer the electrons to this particular compound, and this will change in its excitation wavelength. So it will be purple colored after that. So it will form a formazan. So if this purple colored compound is formed, then in that case, the cell will be alive. Okay, so this is the MTTSA that I was talking about for cell viability testing. Now, second is annexin 5 testing. So in that case, what is the main principle is that when the cells are in complete alive state, so the phosphatidylserine is present on the inner leaflet of the cytoplasm. Okay, but when they are gradually apoptosing, or they are like moving towards the death phase, so what will happen? The inner leaflet comes to the outer side, because the negative charge will be present towards the outer side more. So this leads to expression of phosphatidylserine on the outer leaflet of the cell. So what will happen? There is an XNP that is labeled with FITC or any other fluorophore. So if phosphatidylserine is present on the outer leaflet more, then this annexin V will bind to those, and the calcium ions, in the presence of calcium ions, this will fluoresce. Okay, so this will give us an idea of telling of the cell death. Okay, so how much cell death is there in case of a particular cell culture? So this is an indirect way of assessing the cell viability. Now, the last question is, last point is trypan blue. So what is trypan blue? Trypan blue is a dye that is basically negatively charged. Okay, so whenever the cells is breaking or it is apoptizing, so what will happen? There will be breakage in the cell membrane. So what now, the dye, as it is negatively charged, it cannot go inside to the cell, passing through the cell membrane. So whenever the cell membrane breakage will be there, so this dye will go inside the cell, and then it will give us a blue kind of a cell. Okay, so that means this particular cell is dying, and it is permeable to the trypan blue stain. So this is also an indirect method of determining the cell viability. Okay, so these three I have explained, and there are many more staining procedures or assays that we use for assessing the cell viability. Okay, so if anybody has any doubt in this particular principles, can ask. So this was the last question for the today. And if you guys have any doubt in this particular presentation or in the past lectures, can ask.
So in this particular question, next in 5 and Tricon blue, we are assuming that how many cells are viable based on the cell death. Am I correct, sir?
Yeah.
We are
So that is
looking
why I'm
for
saying
cell
that
death
it is
and we
the
are assuming how many cells.
yeah, it is an indirect way of telling the cell viability because we are calculating the cell death rather than we are counting the cell viable cell or the living cells.
Okay.
So if you guys do not have any doubt, so we'll close the session, and this was the last session for this course. So all the best for the exam, and thank you guys for your patience.
Is it possible to have your email address, my sir?
Um,
Are we allowed to, or I don't know?
okay.